DNA may be analyzed to determine the sequence of nucleotide bases. Walter Gilbert and Frederick Sanger shared the Nobel Prize in Chemistry in 1980 for their contributions concerning nucleic acid sequencing. The Sanger method, or Dideoxy sequencing, is the most commonly used method of DNA sequencing. We will see how to sequence one of the strands. Dideoxy sequencing involves the extension of a short DNA primer by DNA polymerase in 4 separate reactions. Each sequencing reaction contains a single-stranded DNA fragment to be sequenced, DNA primers that anneal to one end of this DNA fragment, DNA polymerase, the 4 normal deoxynucleotides A, C, G, and T, and a small amount, 1% of the total concentration of nucleotides, of a modified nucleotide called dideoxynucleotide. Dideoxynucleotide differs from a deoxynucleotide in that it has a 3' hydrogen rather than a 3' hydroxyl on the deoxyribose sugar. The absence of a 3' hydroxyl on the dideoxynucleotide prevents the formation of a phosphodiester bond with an incoming DNA nucleotide, so DNA synthesis terminates after the addition of this dideoxynucleotide. Each of the 4 sequencing reactions differs by which one of the 4 dideoxynucleotides, A, C, G, or T, is added to the tube. DNA synthesis is allowed to proceed and the primer is extended. In the DDA reaction shown, when the DNA polymerase gets to a T on the template strand, it has the choice of using either the normal A or, more rarely, the dideoxy A. When the dideoxynucleotide is inserted, elongation of the newly synthesized strand stops. Since there are 3 T's on the template, there are 3 places where a dideoxy A can be inserted, and so there are 3 lengths of DNA that can be synthesized, each ending with DDA. For a population of DNA molecules in a reaction tube, new DNA fragments will stop at all possible positions where the particular dideoxynucleotide is incorporated. So, in the DDA reaction, new DNA fragments all ending in DDA will be produced. In the DDG reaction, new DNA fragments all ending in DDG will be produced. In the DDC reaction, new DNA fragments all ending in DDC will be produced. And in the DDT reaction, new DNA fragments all ending in DDT will be produced. Taken together, DNA fragments of all possible lengths covering the region being sequenced are produced. The DNA fragments are separated by size with polyacrylamide gel electrophoresis, and because a radioactive DNA primer was used, the position of the band in the gel is determined by autoradiography. The autoradiogram is then read starting at the bottom where the DNA fragments are the shortest. These bands at the bottom represent the sequence of DNA bases nearest the primer. The sequence is read 1 base at a time in the 5' to 3' direction. The sequence being read is complementary to the DNA fragment. The sequence of this short fragment is 5' A, G, C, C, T, A, G, A, C, T, 3'.